Hello everyone, welcome to lecture 3 of Introduction to Food Chemistry. Today we are going to look at the aspect of purification of sols and a few other aspects under the same topic of sols. One of the methods of purification of sols is by dialysis, and in this method we use the semi-permeable membrane, for example a parchment, that removes small ions. We can also use electrodialysis, and this is where an accelerated dialysis is used with an electric field. Also, ultrafiltration is one other method. Here we the filters are modified to retain colloids, and then pressure is used to speed up the process. What are some of the properties of sols? One of the major properties of a sol is color, and the color depends on the particle size and the wavelength of scattered light. For example, we can have a silver sol appearing, and for a silver sol to appear, the orange-yellow wavelength is the one that will work, and then also the violet at a range of that figure and that figure respectively. The other one is the optical properties. One of the optical properties is what we call the Tyndall effect, and this is the scattering of light by sol particles when a beam of light passes through a liquid. Usually it causes changes in the color of that liquid because of the spread or the dispersion of the light into the different particles, and it causes a visible beam. Ultra microscope. This one also detects particles via the scattered light. And then we can also use the electron microscope, which reveals the particle shape and size up to 10 power 4 magnification. The third property is the kinetic properties. And under this, we have the Brownian motion or the Brownian movement. This is the random zigzag motion of the colloidal particles caused by collisions with dispersion medium molecules. And it is absent in two solutions and suspensions. You can find it in the sols. The fourth property is the electrical properties, and one of these is the particle charge. This property prevents aggregation and hence stabilizes the sol. The other one is electrophoresis. Electrophoresis is the movement of charged sol particles under an electric field. And the third is electrosmosis, the movement of dispersion medium under electric field. Coagulation or precipitation of sols. A sol has a tendency to coagulate or even precipitate. And we have some methods that are applied to cause coagulation or precipitation of sols. One is the electrolyte addition. You can add an electrolyte to the sol, and this electrolyte contains ions that neutralize the particle charge, leading to precipitation. The other one is the electrophoresis, which we already talked about above, and then mixing oppositely charged sols and also boiling. These are methods through which you can achieve precipitation of sols. The protective action of sols. In the previous class, we noted two types of sols, the lyophilic and the lyophobic sols. So the lyophilic sols protect the lyophobic sols from precipitation. If you mix the two sols together, one will prevent the other from precipitation, which is the lyophilic protecting the lyophobic from precipitation. And this is the product that produced after this combination is what we call the protective colloids. For example, gelatin for gold sol. We also have what we call a gold number. This is the minimum amount of protective colloid needed to protect and prevent coagulation of 10 ml of gold sol by sodium chloride. And that was about salts, the remaining part of salts, let us briefly look at gels. A gel is a colloidal system with liquid dispersed in a solid. It forms a semi-solid mass upon cooling, and this is the process of gelation, formation of the semi-solid mass. Once you heat this gel and then you cool it, once it cools, that process is called gelation. An example is the gelatin solution turning into a jelly. You have to note that gels are an important colloidal system, especially in food science, and gelation involves transformation from a sol to a gel. Gels have been applied variously in a lot of processes involving food science and food chemistry, especially in the production of so many products of food. Types of gels. One of the types of gels is elastic gels. This one is flexible and it can regain its shape after deformation. The examples we have the gelatin, the starch, and also soap. The non-elastic gels are rigid and have strong molecular bonds. For example, silica gel. Note, elasticity in gels depends on physical and the chemical bonding. Elastic gels are reversible, and non-elastic gels are chemically cross-linked. That's why they are rigid and they have strong molecular bonds and usually irreversible. Properties of gels. One of the most important properties of gels is hydration. Elastic gels can reabsorb water and hence hydration takes place. Swelling. Volume of gels usually increases when water is absorbed. Syneresis. This is shrinkage with solvent release. Once this water that has been absorbed gets out of the gel, it causes shrinkage. And this is what we term as syneresis. Then we also have what we call thixotrophy. Gels liquefy on shaking and revert when still. We have what we call food gels, the gels that are applied especially in food. Gels in food system have structural and textural roles, and we have various categories of food gels. For example, the plastic, the caseinate, gelatin, polysaccharide, the concentrated, and also the mixed gels. Let's look at the plastic fats. These are fast crystals that are rigid and form brittle gels. The caseinate gels are a result of acid or enzyme induction, and these ones are usually weak and deformable. We also have the gelatin, which is highly elastic and forms triple helices. And then the polysaccharides, these ones contain single or double helices and also have egg box junctions. Emulsions. Let's look at a few basic concepts involved in emulsions. We have emulsions are liquid to liquid colloids, whereby one liquid is dispersed in another. In the previous class, we noted that we have two types of emulsions, which is the oil in water, and the example here is milk. And then we also have the water in oil, which is, example is butter. What is the role of emulsifiers? Emulsifier is one of the most important substances that stabilizes emulsions, and it works by reducing surface tension. For example, a soap molecule. The soap molecule will have the hydrophobic tail and the hydrophilic head. Two important properties of emulsions. One is the emulsification, meaning that this emulsion can change. It can be reversed under certain conditions. And then the dilution test. Adding dispersion medium to test emulsion type is one of the properties that you may find in emulsions. Form. Formation and structure of a form. We say that a foam is a gas in a liquid colloid, and we have a substance that can be added into a foam to cause stability, and this is a surfactant. A surfactant causes the stability of a foam. Foam forms small bubbles that can flatten to form the polyhedral shapes. The polyhedral form behaves like a gel, and yield stress is observed. This one retains the shape under weight. It can be elastic, it can also be viscoelastic. It can transform to elastic to a viscoelastic upon deformation. So this is the basic concepts of sols, emulsifiers, and foams. And this marks the end of our class right now. Thank you so much.